Hello, everyone. My name is Yu Wan, and today I want to present a topic called transcription and translation process. Before we dive in, let's have an overview of how this whole process has been processed. So first thing first, we're inside the nucleus. The transcription has happened inside the DNA, where it has interaction and adding this nucleotide for the protection of the RNA. Which we call a pre-messenger RNA. And because there are many non-coding information inside the DNA, so this pre-messenger RNA actually contains much more information than we needed. And we only want the useful information, which is the exons. And this is a process called splicing. And after we remove the non-coding region, we have immature messenger RNA, and we export it from the nucleus and meet with ribosome. And with assistance of the transfer RNA, we can produce this polypeptide, which is the protein. So now let's first dive into the first process of transcription. So at initiation, a enzyme called RNA polymers binds on the DNA and unwind the double helix structure of the DNA so that the template strand of the DNA is exposed. As the RNA polymers moving along the DNA, where more nucleotides are coming in here to form this RNA transcripted RNA polymers and adding to the 3' end of the growing RNA. And just to be noticed that for the template strand here, it is actually from 3' to 5'. So the direction is from 3' to 5'. So the produced the messenger RNA is from 5' to 3'. And during termination, when the RNA polymers reach to the terminal site, it will release the DNA so that it can left us with a pre-processed messenger RNA. Here, remember that it contains many non-useful information, what we call introns, as shown in here. So the introns here are not really useful for protein transcription, and we want to get rid of it. So this process is called splicing. So the whole process of splicing involves a small protein called small ribonucleus proteins, or SNRNPs. It will first bind to the splicing side of 5' splicing side of the intron and also the 3' splicing side of the intron. The interaction between the two SNRNP will cut the introns from the exons so that the front 3' prime side of the exon, so each group will have a reaction with the 5' phosphate site of the latter exon, so that the two exons will combine together to form this mature messenger RNA. And this is not the end. Before it got export, a 5 cap is added to the 5' side of the messenger RNA, and the poly A tail is added to the 3' side of the messenger RNA. This is mainly to make the messenger RNA more stable, and also as well as helping it for exportations from the nucleus to meet with the ribosome. And before we go to translation, we need to first clear some of the definitions. So first of all, there is a genetic code table where for different genetic coding, it has a corresponding amino acid attaching to it. And this is mainly because of a structure called a transfer RNA, where we are going to discuss it later on in this presentation as well. In this table, you can see that multiple coding can correspond to the same amino acid. However, if you have any unique 3 coding, it will only corresponding to a certain type of amino acid. So there is no ambiguity here. As I mentioned that the matching genetic table is actually from this structure called transfer RNA, where the transfer RNA will bind from the anticodon will bind on the messenger RNA, and each of the transfer RNA will carry its amino acid. So in such a way, we can create this polypeptide. And the place where the transfer RNA bind with the mRNA is in the structure called ribosome. So ribosome actually provides this platform so that the transfer RNA can bind with the messenger RNA and to produce the polypeptide structure. So let's take a look at what the translation process really is. So for the translation process, during the initial process, where a small subunit of the ribosome will bind on the starting codon of the messenger RNA, and a transfer RNA's anticodon will bind with the messenger RNA as well. As you can see here, it carries a amino acid. And later on, a larger subunit will add on to the messenger RNA so that the whole platform is complete. And during the elongation process, more and more transfer RNA, as the ribosomes travel through the messenger RNA, more and more transfer RNA will add into the ribosomes A site. And it will make the previous tRNA will release its amino acids and attach to the incoming transfer RNA so that a multipeptide structure will be formed. And during the termination process, as a ribosome reaches the stop codon on the A site, a release factor will add to the A site of the ribosome so that it will activate to release the transfer RNA along with the processed polypeptide product. So this is actually the final product of the messenger RNA. And later on, the ribosome will dissociate with the messenger RNA so that this will sum up as the whole translation process. So the above presentation summarize how from the DNA to a protein synthesis process looks like, including transcription and translation. Thank you for watching.